Good afternoon to the virtual audience, and let me start by thanking the meeting organizers for the opportunity to provide an update on the status of the H2I2 project. Let me recall you that the goal of H2I2 was to design and assess the expected performance of a hybrid system for beam monitoring and online range verification in particle therapy treatments delivered through the innovative Gatoroid static gantry. The goal was successfully achieved, and we have selected a design based on an ultra-fast silicon detector as beam monitor, which would be installed on the beam line just inside the bore, and an open PET ring that will close the ring inside the bore, and that will provide the signals from the PET coincidences of the prompt gamma emission induced by the beam tissue interactions. The assessment was based on Monte Carlo simulations and was reported, the results were reported in the final report at the end of the phase 1 of ATTRACT. Now, since October, we have focused on the following step, which would be the design and construction of a prototype system. And in order to do so, a crucial issue is the implementation of a data acquisition system. The concept here is the idea of buffering data from the beam monitor and triggering on a signal from one of the PET crystals, which would correspond to either a PET coincidence or a prompt gamma emission. Then, the time tag measured from the trigger signal in one of the PET crystals would have to be associated correctly with the corresponding beam particle time tag, which is buffered in the UFSD data. In order to implement this concept, we have to comply to the constraints, which are essentially the beam maximum rate. The average beam rate at CNAO is of the order of 10 to the 9th particle per second, but the actually measured instant rate, measured with ultra-fast silicon detectors, can reach 10 to the 10th particles per second because of a bunched structure of the beam. To comply with these values, we have to reach at least a 50 picosecond time resolution, which would be possible, for example, by using the Pico TDC developed by CERN, which can provide a time resolution up to 3 picoseconds and a 300 megahertz maximum rate per channel. So the DAC design would be based on FPGAs that will buffer UFSD data and wait for the prompt photon trigger from a PET or prompt gamma module. And then we have to implement an algorithm that provides the correct association. So basically identifies which beam particle in the buffer is the one corresponding to the detected photon. The trigger was simulated with a Monte Carlo study. And on this simulation, we have started to assess what is the best strategy to provide the proper association between the time tag of the detected photon and the time tag of the beam particle. And this algorithm was expected to be tested in a beam test, which was scheduled in November 2020 at CNAO, but unfortunately was delayed because of COVID-19 restrictions. So hopefully we'll be able to carry out this test in the spring, at least as soon as CNAO will be accessible again. And in the meanwhile, we're obviously getting ready to apply to the phase 2 ATTRACT goal. Thank you.
This project is entitled Edge Illumination X-ray Phase Contrast Imaging with Equiangular Time Delay Integration Scanning, EXPITIS. Now, the aim of this project was to combine Edge Illumination X-ray Phase Contrast Imaging with Equiangular Time Delay Integration Scanning to facilitate highly sensitive imaging. Edge illumination is a method that allows conventional laboratory X-ray sources to do phase contrast imaging, and time delay integration equiangular time delay integration is a method of artifact reduction that allows us to get very high quality images. And the advantage here is that it would also eliminate any effects of defects in the masks used for the edge illumination. Since October 2020, we have managed to implement a simple phase retrieval algorithm, and using that we've collected a tomography data set. Now the X-ray flux on this system is very slow, so that's why we haven't collected many data sets like this. This was actually a historic pollution aggregate that we scanned. It's mounted on a Perspex rod using soft wax. And you can see from these two images here that the phase contrast detection is well illustrated by the fact that we can see the Perspex and the wax very clearly in the phase image, whereas it's almost invisible in the attenuation image, and certainly the signal to noise ratio would be very low in the attenuation image. So this really is proof of principle. We have demonstrated that the method works. The alignment is much simpler than it is with the conventional edge illumination system, and also we will be able to scan larger samples and get much higher quality images. So we've demonstrated the advantages of combining time delay integration and edge illumination, but the X-ray flux of this system is much too low for practical use. The scans would just take too long. So going forward, we'd need to demonstrate it on a more powerful X-ray source. For example, the one used by UCL to develop the initial edge illumination system has about 100 times the X-ray flux of the system we used in this prototype. Now, such a highly specialized scanner is combining two techniques which themselves are a bit of a niche, which means that EXPITIS is a niche within a niche, and that would lead to a very small market. So what we would be looking at going forward is a multimodal system where this method of scanning is just one possible mode of different switchable types of imaging. This versatile system we would develop by incorporating features from other ATTRACT projects such as ML Cyclo-CT, but also with other projects it'll be open to anyone else, and maybe we will get some ideas in this kick-off meeting. The most important application is likely to be discrimination of biological samples, but another possibility we've been investigating with different projects in the past is looking at flaws in diamonds prior to cutting. Diamonds having a low X-ray attenuation coefficient produce little contrast, again making them very ideal for use in phase contrast imaging. They're also difficult to image optically because of the very high refractive index. We'll carry on with our cultural heritage work, looking at detection of ink and being able to read damaged scrolls, and we've already got links with Cardiff University for reading damaged scrolls, and also with MIT for reading folded and sealed letters. Both Queen Mary University of London and University College have existing links with X-ray equipment manufacturers, medical and biological researchers, conservationists, and that diamond company. And future sources of funding will be considered in consultation with Queen Mary Innovation and UCL Business to decide the best way forward that would have the best chance of success.
Welcome, this is Davide Deganello from Swansea University. I would like to present the ATTRACT project 3D Synth, commodification of scintillator detectors using 3D printing techniques. Scintillating technologies are an integral component for particle detectors, with wide range of application, nuclear, medical, applied physics. But an issue is that typically these scintillating detectors are bespoke, one-off, expensive. 3D Synth aims to revolutionize this to commodify the use and application of scintillator detectors by integrating 3D printing technology with scintillator materials and electronics. This track project was a partnership of Swansea University and Linkios Technologies. It was successful. 3D printing all these structures were produced, which matching scintillating fibers and electronics, and the prototype was successfully tested, being capable of achieving 2D mapping of elementary particles such as cosmic ray muons. During the evolution of the project, different 3D printing technologies were tested, finally adopting fused deposition modeling for its ability to print internal channels, as shown here in the photo. 3D printing led to the development of a modular holding structure, allowing low-cost and scalable manufacturing. It allowed ease of assembly, joined with physical and light-proof protections of the scintillating materials. Critical, it allowed the ability to precisely guide and improve the position accuracy of the scintillating fibers for better performance. Here, in the images in the slide, you can see the design of the 3D printed modular structures on the left, with a photo on the right of an assembled examples. On the right, there is also a photo showcasing the ability of accurately position the fibers thanks to the new 3D printed solution tested. The assembled structure was tested with a linear scintillating fiber array of 32 fibers. This was directly matched with the silicon photomultiplier array. The operation of a prototype was successfully verified, testing with cosmic ray muons and with a strontium-90 beta source. In the slide, you can see examples of a 2D mapping response for the two cases. Through Attract Phase 1, 3D Synth has been able to achieve technology readiness level 3 to 4. A proof of principle has been accomplished, a prototype has been successfully made and tested. Towards Attract Phase 2, we have started additional collaborations. For example, with BAM, the German Federal Institute for Materials Research and Testing, the European Forum of Eyewear Research Laboratories, and KIME, a leading company on electronic instrumentations. Through Attract Phase 2 and an expanded consortium, the aim will be to develop a detector system for in situ applications at technologically readiness level 5, followed by a full-scale prototype at technology readiness level 7. Main demonstration of our technology in Attract Phase 2 will include civil applications with the study of structural integrity of a wave bridge. Another application will be the imaging in situ of the content of nuclear waste containers. The common thread of these applications will be safety. Safety for nuclear, safety for transport. And this is linked to the society challenges for secure, clean and efficient energy and smart, green and integrated transport. I would like to thank you for the attention. For more information, my mail is here below.
Hello, welcome to this update talk on the Wearable Positron Emission Tomography project funded by Attract, presented on behalf of the team by Stephen Watts. For brevity, Positron Emission Tomography is shortened to PET and this project to WPET. The main partners are the University of Manchester in the UK, Caen in Italy, and the collaboration has expanded to include radiation imaging technologies in Portugal and G4 Associates International in the UK. Current PET systems in hospitals are heavy and cost a few €1 million. They are an important diagnostic tool, but access and cost concerns have prevented the use of PET for early cancer screening. This is the motive for a modular PET system which is portable, low cost, and can be adapted to early cancer screening. COVID-19 access restrictions affected the Phase 1 prototype testing. I am delighted to report that this system has been in operation these last months and we have excellent results. The Phase 1 readout system shown in the schematic uses existing CAN readout boards with significant volume and weight. We have a pre-design of a Phase 2 system using a Temperoc 64-channel ASIC designed for silicon photomultiplier readout that fits within a 2 by 2 square centimeter package. Weight and volume are substantially reduced. The photograph shows the prototype system being tested at CAN. The detectors are positioned on a mannequin with a sodium-22 source fixed inside the body. The two 511 kV photons are detected using two modules placed on opposite sides. The data from CAN was processed by Radiation Imaging Technologies. The 3D image was reconstructed following scans with modules placed in different positions within a 3 by 3 matrix as shown in the video. The figure shows the reconstructed sodium-22 source. The figure area is 30 by 30 square millimeters and the source full width half maximum is 0.7 millimeters and 1 millimeter in X and Y. This is an excellent result, consistent with Monte Carlo studies described in the paper, and proves that one can image well with this type of system. We have concluded from an initial feasibility study that a commercial Intel LiDAR system using 4 to 8 LiDARs can be used to monitor the detector positions in real time. This allows the modular, portable system to be quickly adapted to each individual or application. This slide updates our answers on some key questions for Phase 2. A key conclusion from the Phase 1 project is that we have a system that is modular and scalable. Using 27 by 27 square millimeter modules, we can provide coverage for any PET application and then reconstruct the image. The weight of the system is dominated by the lysocrystal mass. However, we can reduce this further by only using as many modules as required for a specific application. For example, for breast cancer, only the top part of the torso needs to be covered. Moreover, if necessary, we would design a support chair to manage the weight constraint. A modular system also greatly expands the number of PET applications to which the technology can be applied. We have extended our partnership to Temperoc, who have a readout chip with substantially reduced mass and size. We have also identified the Attract Mermaid project as a potential partner. We know the importance of extending our collaboration to include medical physicists and clinicians to provide input to the design, testing and evaluation of the system in a clinical environment. Discussions are underway with clinicians and medical physicists in Rome and Manchester. We list the many applications to which this technology can be applied. Our emphasis is to provide a low-cost, portable system for early cancer screening in any specific part of the body. However, this is a breakthrough development, allowing a modular, Lego-like reconfigurable system easily adaptable to new problems. For example, we could take the system from the shelf and quickly set it up to detect prompt gammas in proton therapy. The last two questions were covered in the paper and brief answers are repeated here. In conclusion, Attract funding has allowed us to confirm the concept that it is possible to make a modular PET system which is portable and low cost, suitable for early cancer screening, which has many other applications. We are very keen to take this technology to the next phase. Thank you for listening. Have a good day.
PRINTBIO represents a new paradigm of biosensors for detecting pollutants by using a fascinating microorganism able to transform metabolism into bioelectricity. Since October 20, we have developed a number of activities that I'm going to summarize now. For instance, we have successfully 3D printed hydrogels loaded with GeoVector which are able to exhibit electrochemical properties. Actually, they're able to produce current. We have also genetically modified GeoVector to control the production of electricity in response to the presence of specific chemicals. So we are domesticated somehow our biosensor cells. We have also constructed electronic devices to remotely transmit the data from bioelectricity to an app cell phone from a remote user.
how are we planning to scale up our achievements and convert them into an industrial product? Well, we have identified 3 different and independent periods in this process. First, the one that we have already finished, almost finished, is the attract phase 1, where we have reached tier 3, being able to prove the successful idea. Then we anticipate at least a couple of years till we reach the MVP, the minimum viable product, and we expect, if we are lucky, to be funded by a track in the phase 2. Okay, after this period we will be able to reach TRL 6. Then we expect at least a couple of years before market launching. Okay, so during this period we expect to reach TRL 8 or 9 and really produce a final product that could be ready for market deployment. So that's the period, the moment where we start the full commercialization, 2024. We are going to need definitely strategic partners in this trip, and we have already identified some of them. For example, we are going to need software and apps developers to make a portable device. That's what we want from our system. Our system should be portable and should be controlled by a smartphone. Why? Because everybody in the world have a smartphone, so they are going to have the tool for playing with our device. We are also going to need expert in molecular biology for developing ribosuites, okay, which is the actual key, the molecular key for unlocking the metabolic circuit in the bacteria to convert the presence of an analyte into a signal compound. There are many SME now in Europe able to provide that service. Regarding material, we wanna be always with the most advanced material having an optimal response, and for that we are gonna play in the graphene-based material field, because it could be also nicely modified, and we are gonna need expert on that. Of course, we need somebody to test our products. We are gonna need end user, okay, able to guarantee that our solution is working. We want to focus on environmental application during this period, okay? In theory, PRINTBIO could be applied to any detection of any analyte, but we want to have a little bit of focus, and we want to focus on environmental application during this period. And last but not least, we have identified and we have contacted at least 3 projects already funded by Attract. We have which are developing a nice innovation, would have some kind of synergies with ours. So we expect also to keep collaborating with them in the phase 2.
Hello everybody, my name is Tobias Schulli, and it's my pleasure to present you our InGaN-FULL-SPECTRUM Attract project and our most recent progress. So in this project, we're tackling a decade-long challenge in material science, namely the modification of the lattice parameter of a crystal in order to accommodate its optoelectronic properties. Concretely, in the case of indium gallium nitride epitaxy, we're seeking to enhance the available indium content in these alloys in order to grow LEDs for all colors of the visible spectrum. The red color is the most difficult one because it requires the highest indium content. If we could expand gallium nitride by up to 2%, that would allow us to accommodate all required indium gallium nitride alloys for these devices. So very recently at the European Synchrotron in December 2020, we have achieved the controlled expansion of a silicon layer by up to 2.1%. So what we can see here in this graph, these are Bragg diffraction curves. So every curve represents a distinct lattice parameter, and we have been able to modify this lattice parameter during observation step by step to fully go to the elastic limit of the material. This has allowed us to achieve a world record level of strain in these silicon membranes. So if we think about all the tools, materials, and the processes that got us there, then we can say they're simple and fully compatible with the very strict requirements of semiconductor industry. And at this moment, we are prospecting device makers as potential alpha customers for such a partnership of scaling up. So beyond the cited example of indium gallium nitride alloys, our process and tools are so generic that they can be applied to any crystalline material to study all the opportunities of strain engineering, which is a vast field at this very moment. And a concrete example is, for example, silicon germanium, where today, although it is a very, very established field on which many technologies are based on, there is still no availability of substrates for all silicon germanium alloys, and their availability could significantly enhance the performances of silicon germanium detectors in the mid-infrared. So at this very moment, we consider our potential participation in ATTRACT-2 as the perfect passport for applying also to private funding for scaling up. And in addition, clearly, ATTRACT-2 should be the opportunity for climbing your TRL ladder from now maybe level 2 to 3 to level 5 to launch a startup. Thank you very much.
Hello, my name is Patrick Steglich and I am from IHP in Germany. I will present an update of our project BioPIC. The main objective of our project is to demonstrate a photonic biosensor in a scalable CMOS technology that is ready for mass production. Here you can see a cross-section of a CMOS chip including photonic biosensors. Compared to standard integration approaches in which the biosensor is released from the top of the chip, in our project we have investigated on the development of a backside release of the biosensor. This allows us for the first time a separation of the sensing area from the rest of the chip for a scalable and cost-effective point-of-care diagnostics. Since October we have focused on the packaging of the chip. First we have tested a new 3D printing method for the wafer scale fabrication of microfluidics, which you can see in figure 1. As a second step, we have investigated on the bonding process of the chip. We need a cold process at room temperature so that the surface functionalization that is realized on wafer level in forehand is not destroyed. So initial tests have demonstrated the feasibility of one process that we have identified since October. Taking all together, our results open a route towards wafer-scale mass production of photonic biosensors. In this way, we make our technology accessible for industry and science, which could result in innovative applications. That's all from my side. Thank you very much for listening and stay healthy. Bye.